Hi everyone. This is mainly a review and a little more detail about the neuromuscular junction. So the neuromuscular junction is the connecting point between the neuron, or the axon of the neuron, and the muscle, or the motor end plate of the muscle. So the neuromuscular junction is that end of the axon, the end of that motor neuron, and the motor end plate is part of the muscle down there. So in general, a synapse is a region of communication that can occur between two neurons or a neuron and a target cell. So in this case, the synapse is between the neuron and the skeletal muscle. So it's called the neuromuscular junction, and it's a type of synapse. So synapses are separated by little clefts or spaces. In this case, it's the space between the neuron and the muscle. So in order for the communication to continue between the neuron and the muscle, neurotransmitters are needed. These are substances found in these little packages here that communicate between the neuron and the muscle in this case, or between one neuron and the next neuron in the case of just neuron-to-neuron synapses. So luckily for the neuromuscular junction, there's only one neurotransmitter, and that's called acetylcholine. So whenever you see ACH, you know that is acetylcholine, and it is the neurotransmitter between the neuron and the muscle. So a little more detail here. So this whole thing is the neuron, and the axon is the part that reaches down towards either another neuron or towards a muscle. So the very end of the axon is a kind of a swollen area called the synaptic end bulb. So the synaptic end bulb is filled with synaptic vesicles. So remember what a vesicle is from Chapter 3? It's a little package. So in this case, there are packages or vesicles filled with acetylcholine, and they're here at the synaptic end bulb. And on the muscle side, the motor end plate is a region of the muscle that's making contact with the neuron. So the special thing about the motor end plate is it has acetylcholine receptors. That's what these little green structures are, acetylcholine receptors. They are there to bind with acetylcholine. So a motor end plate could have 30 million acetylcholine receptors. Okay, so we're going to stop and think here. Do not rush ahead. Make sure you stop and challenge yourself. So the synapse is the whole thing, right? The synaptic end bulb and the motor end plate. So the synaptic end bulb part is this swollen part here. What about voltage-gated calcium channels? Well, we didn't talk about it yet, but there are these purple things here. And you could see, let me erase that, get the pen back, is that these little purple balls are calcium. So these are voltage-gated calcium channels. When a nerve impulse comes down, their gates open and calcium can rush into the neuron. Okay, where are synaptic vesicles filled with acetylcholine? That's these little vesicles here. All these little blobs, packages. The motor end plate, again, was this part here where you see receptors for acetylcholine. The synaptic cleft is the space between the neuron and the muscle. Acetylcholine receptors, I thought we just said that. Did I say that already? Acetylcholine receptors are these little green ones. You can see them binding acetylcholine. And then mitochondria right here. They're always scattered throughout muscle tissue. All right. So here's a little more detail. I'm going to actually go back one and just fill in a part of the story that's not here. And that is the nerve impulse comes down. Let me get the pen back. So the nerve impulse comes down an axon. And when it hits the synaptic end bulb, these calcium voltage-gated channels open because they respond to a change in voltage. Hey, there's electricity coming down the neuron. We're going to open up. And calcium is going to flow into the neuron. And what that does, that stimulates the release of acetylcholine. Just like the drunk guy at the party being kind of staggering around and telling bad jokes. As soon as he walks in the room you're in, everybody decides to leave that room. Well, that's how calcium is with acetylcholine. Calcium moves in, acetylcholine moves out. So that's the stimulus there. So acetylcholine gets released from the synaptic vesicles and into the synaptic cleft. So that's where we are here. So the acetylcholine diffuses across the synaptic cleft and lands on receptors right here. So these receptors for acetylcholine, when acetylcholine lands on them, they open up and allow sodium to move from outside the cell to inside the cell. And when sodium moves inside the cell, a muscle action potential is produced. So it'll travel down the sarcolemma and cause the release of calcium from the sarcoplasmic reticulum. Okay, so calcium is released from the sarcoplasmic reticulum. The muscle cell will shorten and generate force. It'll contract, basically. And then back here at the neuromuscular junction, as soon as the stimulus is ended, acetylcholine will be broken down by an enzyme, acetylcholinesterase. And therefore, acetylcholine can't have the same effect, and the muscle action potential will be stopped. So there's some interesting things that can happen at the neuromuscular junction with various chemicals. So you might have heard of botulism, and you know, hopefully, never to eat food out of a can that's bulging like that. That looks totally dangerous. So this certain bacterium, when it infects your body, it blocks the release of the acetylcholine here at the neuromuscular junction. So if you don't have a release of acetylcholine, you don't have acetylcholine landing here, which means you don't have sodium rushing in, which means you don't have a muscle action potential and you can't have a muscle contraction. So your diaphragm, which is your breathing muscle, will stop. It'll be paralyzed and you'll die because you can't breathe. So here's a picture of a boy with botulism. You can see his whole face is paralyzed because it affects the whole body. It doesn't just affect the diaphragm, it affects everywhere. Curare does a similar thing, but it does it a little bit differently. So you might have heard stories of like these South American Native Americans who have poison darts, basically. So they coat the tip of their arrow with a poison from a plant called curare. And if you get hit with an arrow with curare in it, it'll cause muscle paralysis. This time it's not because acetylcholine is not released. That's not the case, so that's not the situation. Acetylcholine gets released, but the receptors are blocked. So curare blocks the receptors. So the acetylcholine has no receptor to bind to. Therefore, you cannot create a muscle action potential. So curare-like drugs are used to relax the muscle during surgery. So that's a positive use of that particular drug. All right, so one last review of how it all goes. Nerve action potential causes the release of acetylcholine. Acetylcholine binds to receptors. Sodium rushes in and causes a muscle action potential. The muscle action potential travels down the T-tubules and throughout the muscle. So you have that muscle action potential happening everywhere in the muscle fiber at once. So that muscle action potential causes the calcium to be released from the sarcoplasmic reticulum. When calcium is released from the SR, sarcoplasmic reticulum, it lands on troponin. Troponin lifts tropomyosin off the binding sites on actin. Those exposed binding sites are now available for myosin heads to attach. So myosin heads will attach, form cross bridges, do a power stroke, and have a muscle contraction occur, and this will keep happening as long as calcium is present. But eventually, the stimulus will stop, and calcium will be actively transported back into the sarcoplasmic reticulum. There will be no longer calcium on troponin, so tropomyosin will cover up the binding sites again, and your muscle will get to relax. So that's the story in review. Phew. Okay, active learning here, too much listening. You better sketch a neuromuscular junction and label the two main parts. Okay, it might look something like this. Okay, in this case, this is the synaptic end bulb. Oh, I lost, that's supposed to be synaptic end bulb. And this would be the motor end plate. Well, that should be the motor end plate. All right. So, those are the two major parts. The neurotransmitter is acetylcholine. Try to put the following events in the order in which they occur. Okay, the first thing that happens is acetylcholine gets released from the synaptic end bulb. Then the acetylcholine diffuses across the synaptic cleft. That results in activation of acetylcholine receptors, which essentially are sodium channels. So the sodium channels open. Oops, that should be 4, not an N. I'm so excited about sodium. Okay, then that causes the production of a muscle fiber action potential. So eventually everything ends at the neuromuscular junction when acetylcholine esterase breaks down acetylcholine. So on the exam, you may have activities like this. Put the steps in the order in which they occur. So it could happen for the neuromuscular junction. It could happen for the contraction cycle. It could happen for, I'm trying to think of what else, the troponin, tropomyosin, calcium connection. It could happen for any of those. So when you see physiology, make sure you put things in order and understand the order in which they occur. And again, the animations are great for that. So make sure to just view the animation on the neuromuscular junction. Okay, just finally, the EMG, you probably did this in lab if you're taking lab. It measures electrical activity, the muscle action potential in resting and contracting muscle. So resting muscle looks like this. There's no electrical activity. A little bit of contraction might look like this. A big contraction, like you're squeezing hard on a hand grip, might look like that. So the electrical activity reflects, or I should say the EMG reflects the electrical activity in the muscle. So these are called electrodes, but in some cases you have fine wire inserted into the muscle. So these can be used for diagnostic purposes. Okay, thanks for listening, and please let me know if you have any questions.